But in any case, I'm going to talk a bit about Norway rats, their ecology, and how the ecology relates to what we know about leptospire as a pathogen of Norway rats, Rasnomegicus, and as a major maintenance reservoir. And I'm going to talk mostly about information that comes from Baltimore, where rats have been studied in their association with leptospire since since 1917 or 18, when it was first described in rats from Baltimore, not just years after the first description of Norway rats as a reservoir from Japan. In any case, oh, okay. Acknowledgements, usual cast of characters. I'm not going to go through all of them, but many people from Yale, many people from Field Crews, our UK partners, and down in Liverpool, because Peter's sort of probability cloud. And in any case, I'll try and refer to these people as we go, but I won't get all of them in. This is not Peter. This is the critter of interest. It's Norway rat, Rasnomegicus, and we can actually reconstruct basic information about how this species got around the world and when it arrived or successfully colonized certain areas such that it was noticed. And during the 1800s, it's red. If the box circles half filled, it's about 1850. You can see that often by the 1700s, rats were distributed not only in the Old World, but it also got into the New World. Fairly recently, these rats, that Norway rats, were found in Britain. I think it goes back to the early 1700s or mid-1700s. So these are recent colonists. And it's clear from some work that we've done, I think from others, that along with their migration or immigration with trade routes, these animals have carried with them certain pathogens. And I think leptospira, pterigans, Copenhagenii, and hemorrhagia are going to be clearly one example. Sole virus, we know, is distributed wherever Norway rats are found. Sole virus is a hantavirus, first described in Korea in about 1981. It's different from that because of hemorrhagic fever. And we also know that various species of Bartonella are disseminated with them. We can find these species throughout the world at different frequencies. So these animals have carried with them many pathogens which are problems. Now, you can't talk about rats without showing some habitat pictures. Albert showed them yesterday. I'll go through these fast. In Baltimore, slums are blighted often by many vacant buildings. This is not good habitat for rats, but when we have interspersed of it residences which are inhabited, this can lead to ideal conditions, because in these neighborhoods, sanitation is let's just say inadequate, and often the persons involved who are living in these neighborhoods are not taking great pride in their living conditions, and so refuse builds up, as opposed to slums in Salvador, where there are vacant buildings, but these are relatively high density, low SES conditions. So if we look at Baltimore, this is an alley, here's a young man. You can see that in some of these areas, huge accumulations of garbage occur. And in Salvador, proximity to some of these large dumps has been associated with risk. I think that that's a likely case. And Albert showed you some pictures of burrows in Salvador, which I'm not going to repeat. But these are rat burrows. And I think you can appreciate the huge amount of dirt which is being excavated here. You can see numerous and the amount of dirt excavated, you can see the crack here. This whole cement structure, backyard, has been split in half by the amount of dirt which has been excavated among it. So there's structural damage as well. In areas where there's not concrete, you can see these massive accumulations of dirt where these rats are going in along the side of alleyways in Baltimore. Mike showed you the model yesterday, and what I'm going to try and get at is some of the parameters which are in the model for which we need estimates and how we may provide those estimates. And this is a process which is imperfect, and I'll point out some of the problems. We're interested in leptospire sheddings kappa, how much how much leptospire is shed per rat per day. We use genome equivalents. The role of vertical transmission, U1, sexual transmission, U2, or other horizontal means of transmission, and environmental transmission. Of course, this plays a role in probably in rat-to-rat transmission via the environment, in addition to being the major component in how humans become infected. We've talked a lot about the environment and how to link it to models of transmission in rats. And as Mike mentioned yesterday, the number of leptospires and the U3 may be the proper way to look at the force of infection. And I used to use this term dynamic demography. Why is demography important? Why is population data important in terms of leptospirosis? And this is data which just came out in 2014, published by Federico Costa and others. And what I want to show you is, we use two different methods to detect reservoirs. Leptospires, both isolation and in this case, coronal sections of kidneys staining by an IFA technique. And they both gave identical results. Essentially, leptospiral infection is fairly high in these youngest classes. These are rats under 200 grams, generally not sexually mature. Both males and females tend to become sexually mature by any measure around a threshold of 200 grams. So these animals quickly acquire leptospirosis. In excess of 70 to 80% are infected by the time they reach adult. If you look at other infections, I'm just pointing this out, that the dynamics of infections in these populations is highly variable. It depends on what you're looking at. If you look at sole virus infection, it's one of these, and there are two studies on that. Very low levels, appears to only come up rather moderately, and we only have little data on Bartonella, but this shows the acquisition or the infection of Bartonella also among these rats. So co-infections are common. Now look at the same data from Baltimore. The sole virus is the triangles. I'll just take that first. You see extremely high levels of infection, approaching in some areas, well, in some areas 100% of adult rats. Again, the same category. These are exact same categories that we're using in Salvador. Less than 200 juvenile, 200 to 400 subadult, and greater than 400 adult. Somewhat arbitrary, but there are reasons why we use these breakdowns. Now take leptospire interrogant. Juveniles are infected at roughly around 15%, males and females. And that might be less than half or a third of what we're finding in juveniles in Salvador. Now, remember, these data are preliminary from Salvador. Whether this will hold up, but a couple of different studies have shown this high prevalence. And then you see it goes up and reaches an asymptote, somewhat close to Salvador, but Salvador young adults are much more similar to the large mature adults, and so that the patterns will be quite different. Preliminary. Again, why does demography, and we're using mass throughout these areas as a surrogate measure of age. In fact, in a number of studies conducted by Norm Slade some years ago, mass can actually be a better indicator of age than many other measures. So take it for a grain of salt. But one of the things which has been found in two different studies by us in Salvador is this excess of females in the lowest juvenile classes being infected compared to those in males. Rapidly thereafter, as I said, comes up extremely rapidly and reaches the asymptote about 80 in subadults, adult populations. Again, a different pattern than what you might see in Baltimore. We aren't sure of the differences. In Baltimore, they found no differences in prevalence based on sex. We'll have to see how this holds out. But we also have unpublished data which shows the exact same pattern in additional number of rats. But clearly the size, the age of the animals is important. Yeah.
What do you know of sexual contacts in the different age classes?
We know quite a bit about sexual contacts after the adults are receptive. Female may only be receptive for a single evening, but in some studies you can see over 1,000 mounts that one evening. And although there may be a male chaperone, you can't fight them all off. No, they may have a dominant male. Frequently in colonies will have a dominant male who do their best to protect the female, but when you have a train of males following you, you can't fight them all off at once. So it's intense while it lasts. Let me put it that way. Not that we know of. Now, I'm going to come to that because some of the interesting findings, remember, much of what we know are from temperate cities. We know virtually nothing about tropical slum Norway rat populations. So when we use some of these metrics, which are derived from temperate rats, and apply them to our tropical rats, we aren't sure of how, if they capture the same information, or whether they'll be of the same use. So this is a work in process. Now, I'll come to that question shortly. And just to show you one of the things, which is, these are two independent variables. And I'm going to come back to this. Body mass is clearly an indication of risk factor. So there's an increase in probability of infection per gram. And mean scars, not significant, but I want to show these data, highly significant by bivariate analyses. This has been shown to be a huge predictive variable for not only infection of soil virus, but we've shown that in longitudinal studies that wounding between periods of recapture is highly associated with development of an incident case we can isolate. Not to belabor this point, we have no idea what goes on in demographic shifts in Salvador over time. This is just an illustration of data which we put together on rats in Baltimore, males and females. And what you'll just note in these lowest categories, they're pulses in which we tend to see young born and decline. But you can see that the demographic profile of these animals is quite different over time. And since shedding is related to demography, we need to know a little bit of something about how these parameters change. And I'll show you again when we come to actual amounts of leptospire shed that this is an important variable to consider. And here's the reason why we're concerned, in part, about using metrics derived from temperate rats to inform us about demographic structure in tropical animals. This is a paper which was in press in the Journal of Mammalogy, just got accepted. And if you look at length and body mass, which are two of the most common characteristics or features of animals, which are used to measure their size or their age, the red dots are a matched sample of rats from Baltimore, matched by slum environment, matched by season of capture, both corresponding to winter, and other variables which we think are some of the best variables which have been used to look at, say, something like Bergmann's rule, which normally doesn't take into account location or time of year or other features of animals which you might want to stratify on, such as the age. But in every category, you see that the Baltimore animals are significantly larger, longer, and have different features based on this one comparison in one season. Now, what this suggests is possibly that using the same sort of metrics that are derived from temperate populations may not be appropriate, but we don't know enough about it. We don't know anything literally about tropical slum populations of Norway rats. No idea for Salvador. This is cumulative mortality. It shouldn't say that. It should be cumulative loss to the trappable population of rodents. I can't tell you how much work went into putting together this sort of slide. Rats are incredibly intelligent, and trying to recapture them is not an endeavor which I would invite you to do, especially when you're doing this in urban areas. catch your rats, you go home and you pick blood and do everything. Then you have to return them to the urban area where you caught them, exact site. You know, and people are very understanding when you're killing the rats, but you're only borrowing them. So I wouldn't try and get this through an IRB committee today. But what you can see in two different habitats, urban and parkland, this is actually ways to go, we see about 50% of the population disappear from the trappable population by a couple months. And once you make it past that, you know, you've got a pretty good probability of making it to ripe age of over a year. So not particularly long-lived, but again, these data are truncated. We don't have all the information. Extremely hard data to collect. And another parameter, which is obviously important when thinking about contamination of the environment, you should have an estimate of whatever you want, abundance. I hesitate to use the word density, but I think you can in some situations. But some sort of estimate of the number of animals which you're dealing with. And of course, with that, you want to know something about their structure, the direct measures, and there are also measures which we can use which can complement these in terms of genetic diversity using fingerprinting methods, which should allow us to get at least a handle on the breeding population. This is the simplest method. I'm only going to show you a couple examples. One from Baltimore, one from Salvador. Here's the cumulative number of rats which are caught. So on day 1, you might catch 10. Day 2, you might only catch 2, but you're removing these animals. And what you see is that as you move more, the cumulative number of animals which you remove approaches an asymptote. And from that asymptote, you can glean some measure of the estimated trappable population to some extent. Trap avoidance, neophobia is a huge problem. And this just shows another example from Baltimore using the same approach. Now, we've actually used a number of methods, and I think, Mike, you talked about this yesterday. And the removal method, you can use catch effort, which is similar to removal method, except you're summing the number of the effort, the number of trap nights that you're putting out over the period, and then looking at the relationship. And this is the model which Amanda, I think, and Albert, Peter came up with. I'm not sure who contributed what. And it looks basically at the probability of capture in days. And based on just, again, limited information, the confidence intervals, at least around our estimates, appear at least nice. And if you have any questions about this, feel free to ask Mike. So abundance is important. We're also doing work, Amanda and Kate Hacker, looking at another measure of rat abundance. These are tracking boards, and yes, this is a footprint. You see other things which can mask rat activity. You have to censor some of these panels, like the cat footprint, but you can fairly objectively determine the proportion of tiles which are covered with some evidence of a tail slide or something which belongs to a rat. So all these are talking abundance. Some of them can get movement, but movement is obviously extremely important. How far these animals go? What's the importance of essentially adjacent communities, say a rate of mu of rats coming from just adjacent communities, which some of our valleys would approach, and also say a lambda, the risk of a long distance translocation of an animal or a migration animal. What do we know about rats movement? Not a hell of a lot. I put this up because it illustrates a study, again, some of these best studies were done in the 1950s in Baltimore, and looks at the movements of males and females. The majority of animals in this sort of valley situation are moving less than 10 meters. This is an alley. Frequently, this is the area where I showed you those rat burrows. Water in these alleys are streets. And streets are effective barriers to rat movements in Baltimore. Now, I'll show you some little data that shows that. But we don't know what could restrain rats in Salvador or other areas where there aren't obvious, say, physical barriers we know which might constrain the movements of animals and limit their probability of venturing forth. Again, data from Baltimore. This is genetic data. No one talked about genetic data at this meeting. When you're looking at gene flow or immigration, you can get some idea of how communities, neighboring communities interact, and the probability of animals coming into a certain genetically classified population from outside, and how far and how connected they are. And this will get back to the whole idea of an eradication unit for control of Norway rats. In this particular slide, you can see that in 1797, the Port of Baltimore was established, and where you see these clusters, those are genetically related populations that share many of the microsatellite alleles that were tested. And then as you can see, as you advance out, 1816 to 1918, you tend to see differences in the profile. Absolutely imperfect data, but you can see that there is genetic structure. This is fascinating. Jefferson, Luzerne, and Jefferson, forget what the street name is. When you look at populations in two alleys, separated by one street, you can actually classify these animals at the level of an alley. So the movement, the restricted movement is real. Not only do you have marker recapture data, but you also have genetic analyses which indicate that the isolation of rats within these single blocks is sufficient to cause at least modifications in frequency of various alleles, so you can even use that at a very fine scale. And I'll show you, we've used it in Pau de Lima, work by Gisela Cacconi and her students. Looking, I think it was 16 different microsatellites. Various microsatellites were redundant, gave us similar information. So that moved into these color curves. Each line represents a single rat. The number of alleles add up to 100%, obviously. And if you look at sites, these sites are close. These sites are separated, they're same valley, they're separated by really an area of about 50 meters to 100 meters of brush where people don't have their houses. You can see some significant genetic differences. And as you go farther out, this area is about 2 or 3 kilometers separated. And not to belabor the point, but we can see by looking at an animal, for example, here, this is where this animal originated, but he would be assigned to this genetic group. So what we're looking at is gene flow. What's important here is we're looking at immigration and immigration pathways. How areas are interconnected? What's the probability of immigration from sites which may be adjacent versus sites which are farther away? And so what we're seeing is good evidence that these rats do move. We had 5 of these events that were picked up. I think 4 of them were males, or maybe more, but anyway, they're dominantly male. The ones in Baltimore, they did not distinguish. In Baltimore, they found rats which had moved over 4 miles. You know, but catch a rat in a garbage bag and close it up and throw it on a truck. That rat's going to be transported. It has nothing to do with his walking abilities. So rats get around a lot of different ways, but irrespective of the method in which they travel, they can introduce new genes into populations which have profound effects. Okay, let me go on. This is a picture of a coronal section of a kidney that's just pressed on a slide, lysine coated slide, and stained by an IFA technique for leptospires. People ask today, can you look at the association between leptospire presence in kidneys and leptospiral shedding? And these are measures of genome equivalence, and you see that these axes should be, all this data is brand new, the axes should be switched, should be that. concentration in urine as a function of kidney. But you can see you can count for a very high percentage of the variance in these two very different compartments, simply by looking at the genome equivalents. These are, for biological data, this is pretty good.
This one natural infection or?
Natural infection.
Oh.
Taking urine from animals which are euthanized, taking kidney, and looking at weight per volume samples. And you can also see that sometimes you're getting 10 to the 8th plus leptospires per ml of urine or kidney. I mean, these are impressive titers. That's per ml. Looking again, and this is one of the things which I think is interesting, again, grain of salt, this is preliminary. We didn't see much PCR positive in rat kidney, again, quantitative PCR, and rat urine. Not much difference, male and females, virtually identical titers, both. What we did find, if you look at the weight categories, if you look at the rat kidney, juveniles have a lower concentration on a log scale, and when you look at the urine, it's even more apparent. So these, not only is the prevalence of infection varying among these demographic groups, but the amount of leptospire that they're shedding in the urine, at least measured by this way, also varies. We found significant differences between sites, not only in terms of prevalence, but also in terms of when controlled for by all the other variables, the amount of leptospire shed in urine. Why, we have no idea. Ratio length to weight. Measure of quality or healthiness of the rat. Totally imperfect, but as this increases, the rats become less healthy. And what we tend to see, and this was significant in some of the analyses, that the healthier the rat, the more leptospires are shedding. All right. The healthier.
How do you judge healthy?
By the length-weight ratio.
Oh, the length, okay.
So if you're 100 millimeters and you weigh 50 grams, 4. If you're 100 millimeters and you weigh 100 grams, it's 1. So, and the other thing, yeah. It's one measure. The groups in Vancouver are using a body fat measure, which may be an improvement, but we're not using that. We can easily adapt. Yes, that is a common measure. To move on sort of rapidly, I've mentioned this in the past. If you look at the microglutination titer, that in rat kidney, as the glutination titer increases, so does the concentration of leptospire significantly. As the microglutination titer increases, the leptospire concentrations in urine increase. Paradoxical? Not necessarily. Not when you have a chronic carrier, might be systemically boosting, or periodically boosting itself. Antibody titer has, in this case, no impact whatsoever on carriage or shedding. Scars, very interesting phenomenon. You see increases in leptospiral shedding associated with increasing scarring, as well as in the concentration in urine. Now, this scarring can be confounded with mass. It happens more often in adult males now, females, but these remain independent risk factors in the multivariate analysis, which I'm not going to show you. And why is this important? I think some of these animals are really messed up. Primary route of soul virus. Now, we don't know all the ways in which leptospire are transmitted. I'm talking about a couple, but there have been reasonable number of reports about direct transmission from an animal bite. Now, leptospire is present in our pharyngeal swabs. They're present in buccal cavity. We have no idea, but we have samples, so we're going to look at this phenomenon and see that if this type of interaction, aggressive interaction, can lead to transmission. Okay, vertical or neonatal, one other of our transmission parameters, trying to get a handle on it, very difficult. we've looked at a number of ways. This is simply taking a pregnant female who's lactating and gently massaging her breast, not really, and putting a drop of milk on these polylysine slides, and then looking at the red fluorescent antibody. And you can clearly see leptospires here. However, these samples are collected in the field, and there's also a problem of contamination of the skin, body parts with urine, and so some of these may not be very convincing. So we went on and did other studies. This is a immunohistochemical staining of one of the pathogenic loci protein in a mammary gland. And this I actually read as milk. And in it you can clearly make out even the spirochetal formation of these leptospires present in the mammary gland. They're there. And proof of pudding. Oop. Albert showed you one of these slides yesterday. This is mammary tissue. Scanning again. You clearly see not only leptospires, but what appear to be biofilms in the mammary gland. Now, what does this mean? I don't know. It's there. Is it contributing to transmission? Something that we don't know. These are obviously suggestive data. We don't know anything yet about transplacental transmission. And when you look at the possibility of, say, oropharyngeal secretion of leptospires, with supporting evidence that rat bite can actually transmit leptospirosis, but in many ways you can contaminate your mouth. One of the things which would be very interesting to look at on some of the things is the potential for early juvenile, mother-to-juvenile transmission of leptospires, because female rats in their burrows were responsible for cleaning all the waste products from their newborns. They lick them and they ingest all the urine, feces, and whatever comes out of these newborn rats. And that involves licking certain tender mucous membranes, to say the least. And it's possible that if we can find these leptospires in oropharyngeal swabs, there may yet be another horizontal or neonatal mechanism for transmission. We haven't looked at that. Nobody has. This system we know, we know more about what we don't know. This way that we go on the grand scale. And the questions are still there, but perhaps easier to deal with. This is, again, Baltimore after a rodenticide campaign. You know, we couldn't coordinate with the city rat control program, and they just knocked out a perfectly good rat alley. Not for long. After a warfarin type treatment, animals are coming out of their burrows, they're dying in the alleyways, you can count them. So let's go to Baltimore. Baltimore has been, like many cities in the United States in the 1950s and continuing on, well, in the early 90s, there was a program by CDC called the Block Improvement Program. And they were giving money to cities. They gave them all these urban rat survey methods. People go out and tally the number of feces, rat trails. But in 1952, David E. Davis published a paper and looked at the number of rats per human dwelling. And this is low socioeconomic areas, same thing today, right around Johns Hopkins Hospital. And this was repeated in 2004 using very similar methods to what David E. Davis did in the 1950s. It suggests that there has been no change in the rat population in terms of numbers, and that rodent control efforts over the last 50 years have been totally ineffective. So we talk about Norway rats and control. What are the data?
We don't know what would happen if you had done rodent control.
No, we don't, but we do know what...
We know that it hasn't, like, improved it, but
It could.
You know, I mean, it could have gotten a lot worse if you didn't do that.
It could, and you know, people when they think about rodent control, they sometimes think about predators. Remember that cat out there to kill all the rats. These are photographs I took in an alley in Baltimore. They show typical interactions between these two. Actually, cats do kill some rats, but it's size dependent. They're only killing the really weedy ones, which aren't effective with anything anyway.
But does it have like a manipulation of the host? I mean, that's what like
Very-
Gandhi does, right?
Exactly. I was just going to say that. There's evidence Toxoplasma gondii can change the behavior of rats such that they lose their fear of cat urine. Whether that's happening here, I did test these rats for Toxoplasma. And a rat this size, virtually 100% infected. But that rat's not going to pass on its Toxoplasma to that cat. Anyway, sort of fun. So what happens when you have perfect control of an alley? In one area, perfect control. And this is work again by David E. Davis, who did everything in the 1950s that I can imagine. We were shadows following him. But they knocked out the population. They trapped before, then they poisoned the hell out of his block. And the man was great. He had a couple of years where there was nothing. They couldn't catch anything. These were sampling points. And then suddenly, boom, rats appear. And look how rapidly these populations grow. I didn't mention fecundity. The median size litter for fetuses in Baltimore is around 9. It varies with size. Theoretically, they go into postpartum estrus. So after a period of about 22 days, in theory, they could breed again right away. In fact, they don't. Studies again by David E. Davis, the incidence of littering is about 7 per year. But when you think about the number of pups which they're throwing out, you can understand this sort of element. And once again, it seems to have maybe settled down to an equilibrium. Not did that for years. That's damn good. This is more recent data by Greg Glass, and they actually estimated the population size. These were in alleys. These fixed units in which there's very little emigration or immigration. They tend to be closed systems. And he tracked and estimated population by a couple of different methods, and then went back and trapped them about 6 to 12 months later. And what you'll note, that's the original, that's 6 to 12 months later, the populations are back. And what percent did he pull out? He was estimating he pulled from 20 to 40% of the population out. Again, not driving it to zero. But rat eradication and control is remarkably different. We know from genetics, a few studies done on islands, that the populations which grow up following control efforts are composed not only of residual populations based on microsatellite data, but you can also see immigration into these communities, at least in some situations. So both play a role in the recovery of populations. And as I mentioned, looking at genetics and other structures, we can hopefully put together maps of connectivity where similar genetic profiles are linked, where we can find levels of immigration and help define a eradication unit that makes biological sense, rather than simply dumping bait in one block and hoping that it's going to be effective. So we don't know what the eradication unit is. No one does. But I think that's something which we can possibly get a handle on. Thank you. I'm thrilled.